Okay, so everyone, thank you very much. Thank you to Andrea and Matt for the opportunity to give uh, I guess, a research update. Let's see here. Uh, to really, yeah, talk about our plant pathology research. Um, And before I begin, I just want to say I'm very fortunate to work with an amazing team. Thank you very much to all the growers and producers that we interact and work closely with. I have a fantastic laboratory team. You see their faces, all committed to doing such a great job. And the unit, the center and beyond, you can see that it is a multi-team effort. And so for today, I'd like to really just kind of get on the same page as far as some of the plant pathology definitions, talk about really briefly how disease is caused, get into these host pathogen interactions that we've been working on in the last, maybe couple year updates, and then give an overall summary and talk about some of our future work. Okay, and I know this is going to be a lot of information. I have a lot of pictures. I figure it's a good record, and please, feel free to reach out and ask for more questions or more info. But we're going to talk about macadamia, cacao, coffee, and soursop, and Annona species today. When we talk about plant disease, you have to remember it's an infectious or biotic process, so it occurs over time. And we're always trying to figure out, well, if something looks strange, do we know what it means to look normal? And then you'll hear constantly some of the definitions of a symptom, which is the host response to the pathogen, and a sign, which is the physical presence of the pathogen in the host. And we may see a lot of the same, say, fungus, but until we prove pathogenicity by conducting Koch's postulates, we can't claim that to be the primary pathogen until that process is proven. So you can see here, we talk about and work with every day the disease triangle. In the simplest terms, for disease to occur, you need to have a susceptible host, you need to have the pathogen present, and you have to have the favorable environment. And that seems very simple, but it's actually quite complex, especially when you're trying to recreate it artificially. We're trying to understand a disease cycle. Where the pathogen survives, how it spreads. So we can manipulate, hopefully alter the cycle to ultimately figure out when we know what pathogen we're working with and how to control it. My team essentially, we look at what it is, how bad it is, and how we can manage it. Okay, so to begin with macadamia. We've been working with macadamia. I mean, it's hard to believe, for more than almost 20 years, working with macadamia quick decline, which is very unique to the state of Hawaii. Quick decline is caused by two Phytophthora species. Originally, it was caused by Phytophthora tropicalis. We understood what was going on. The symptoms and signs, as you can see here, trunk bleeding, then the stressed trees attract ambrosia beetles. And inside, here's the sign. So that's the physical presence of Phytophthora causing discoloration in the wood. And working with varieties and what might be resistant or tolerant, getting a handle on how to manage and what fungicides to apply, how and when. MQD kind of died down for quite some time. Then we saw an emergence or a re-emergence of MQD almost a decade later and figured out that it was a second species of Phytophthora causing the problem. And you can see here, this is the microscopic fungus. They do look different, so we knew right away it was different, but we went through and proved pathogenicity by completing Koch's postulates, just this fungus on healthy seedlings to recreate the symptoms we were seeing in the field. And you had to know what it is in order to manage it. And I will mention there is another species causing very minor problems in Hawaii, but major problems throughout the rest of the macadamia growing region throughout the world, Phytophthora cinnamomum. We did detect it actually just in one location, probably in all this time we've been working in a new planting in Kau, but it has the capacity to cause real damage as well. Okay, so some of the recent advancements, as I said, we need to figure out what it is and how bad it is. And we typically either get samples sent to us or we go and collect samples. And we do have a vast capacity to test and detect pathogens. Sometimes that's microscopically, sometimes that's through molecular techniques. And oftentimes, because we've been studying MQD for so long, we have a rapid test that we can utilize to see if it's one of the two quick decline pathogens. But we needed to hone in precisely and improve sampling techniques, which was really seeing what kind of sampling we do and how least invasive we can get, particularly if we can just use a small drill bit. How far into the wood do we need to go? So we did a lot of comparisons and figured out that shallow sampling was actually a much better way of sampling to get positive detection for the molecular test. And I always like to say it is one thing to confirm that your pathogen is present in the sample, but is it viable? Is it alive? Because just because there's DNA there, well, maybe it's not living anymore and you don't have to manage in ways you might think. So we also devised a new baiting technique to determine viability of these wood samples of macadamia. And traditionally, an apple was used where you could get two samples and it would take a number of days. We actually constructed a new detection method where you could get more than 100 samples for each apple with an improved success rate and a shorter time to get viable cultures. Okay. I mentioned varieties, and we do a lot of variety screening. We're very, very fortunate to be part of the germplasm repository here for macadamia. And so the team, we devised an assay to rapidly test germplasm to look for resistance to Phytophthora. And so we look for resistance in 4 species. So the 3 I mentioned from 2 from macadamia quick decline, the cinnamomum, and actually a 4th species found in Hawaii, not as prevalent on macadamia, but still present. And essentially going and collecting nuts, growing up small seedlings, conducting a seedling bioassay. So in a number of weeks, we can determine if further testing is warranted for some of our varieties. And you can see here that definitely it's kind of color-coded, but it is dependent on the species you're working with, which is very important. Some of these are broad host range pathogens, some are very specific, but these are all potential macadamia pathogens. And certain varieties here fare much better when it comes to incidence. So this is good information as far as available rootstock material or potential seedlings, varieties, because most of the commercially planted varieties here are very susceptible to macadamia quick decline. So we're starting to get a better understanding. And this is also working with UH and the variety trials conducted by Andrea and others and material at UH. So we're really working hand in hand with the industry to see what we can help for new tools. And sometimes a tool in toolbox is chemical control. So we're also working with the university doing testing of fungicides. And this is plates. Essentially, you just put some of these products in agar plates, various concentrations, and it's important to test against the pathogens that you're looking at. Here, 3 species of Phytophthora, just to give you an idea, and the various chemistries, whether or not they're registered from academia, how to apply them. As Andrea always says, the label is the law. You have to follow that. But to get an idea, you can see, first of all, that the fungi look very different depending on species, and then how they react to various concentrations are different. You're ultimately trying to find something like this where there's no growth, but even better if you take this plug, put it onto a media with no chemical and get no growth to see a fungicidal effect. So some of this new product, which actually IR4 is testing against Phytophthora in cacao, might be a new, something new for the industry, and that's ongoing work. Okay, deep breath. Next host, cacao. The industry is really going gangbusters. Of course, the more that's planted, problems, you start to see various diseases. We do have certain things here in Hawaii. There are far worse things elsewhere in the world. Some are viruses, and it was unknown if viruses existed in Hawaii in cacao. So we did an extensive screen testing not only the germplasm repository, which serves as a backup for all of the cacao germplasm that's housed in Puerto Rico, but also at commercial farms in East Hawaii, West Hawaii, even on Oahu, looking, trying to define symptomology of some of this chlorotic suspect viral samples and see if it was, how widespread it was. And looking at, feels like close to maybe 800 or 900 different samples, we found a 20% detection rate, really meaning that it is well distributed on island, particularly. These viruses are vectored by mealybugs. And part of the study also was for the first time determining that plants in the Malvacearum, the malvaceous hosts, could actually serve as an alternative host. So not only is it well distributed in cacao, and actually it doesn't appear to be doing anything to production here or really elsewhere, but things like hibiscus and durian can actually serve as an alternative host for the viruses. So if these are planted close to where cacao is planted, it is possible that these mealybugs can go back and forth and continue to spread the virus. Okay. A lot of what we do, we study things that are emerging or potentially re-emerging on crops. And when we were conducting the giant survey for cacao mild mosaic virus, that's the only one that we have found here, which is, again, a rather mild cacao virus. A lot of the growers and what we were noticing, there seemed to be this slow decline or dieback occurring on some of the trees, even at the germplasm repository. Here are some of the symptoms. And so we conducted large surveys and sampling, giving it a rating scale based on incidence and severity. Ultimately, although some of these pictures look pretty harsh, overall incidence and severity was rather low overall. We did isolate a number of fungi, which these are considered endophytic. So they survive in trees, and when trees become stressed or wounding occurs, sometimes these pathogens can cause some of this dieback. And this is well known for cacao and lots of other crops here grown in Hawaii. But we were just curious, this increase now in incidence and severity. We did conduct pathogenicity studies after quite a number of months. We really did not see any outward symptoms, but when we looked inside, we could see the sign or the presence of the pathogen extending maybe slightly beyond the inoculation point. So yes, they potentially could contribute to a problem, but overall, what's been going on here is a lot of areas, it's a bit of lack of water, and particularly Queensland longhorn beetle. So more of that as opposed to really an emerging problem caused by fungi. What we're moving into more, and one of the top priorities of the industry, is pod rot. And so we could say the reemergence of black pod caused by Phytophthora species, but interestingly enough, some of it looks very different. Some of the symptoms. So we want to make sure it is the same thing and maybe the same, definitely the same species. But in addition, we see other symptomology, and we need to get a real good handle on what that is, especially when it comes to management and potential fungicide use. Okay. The group does a lot of work with coffee. And some of the major ones, yes, we have coffee leaf rust present now here. We're the last coffee growing region of the world to get CLR. But we had been suffering from a number of leaf spotters for quite a number of years, which tend to be, I guess, well handled by growers. And I'd say in the last few years, there seems to be what is considered reemerging. So anthracnose, cercospora spot, there are records. They are here and they had been pretty well managed. And the spots would tend to be maybe smaller in nature, but what we're starting to see is much larger spots, causing almost blight. And when we were doing a bit more digging, we realized that there wasn't a well-defined what pathogens were really causing. It's a lot of information and a lot of information from around the world, but precisely what species of Calototricum and a better ID and understanding of Cercospora. So this is some of the areas we're moving into to actually characterize truly these diseases, what genera and species of fungi are causing them. And we're also partnering with Dr. Lovelace of ARS in Beltsville to do some of the first genome sequencing of these pathogens to really better hone in on the species ID and the genetic variability. And we're very, very curious if there's any fungicide resistance. Is this potentially why the problem seems to be getting worse? Or is it new species, or is it environmental? So you can see we have our challenges, but we're hoping to help answer some of these questions. And looking at some of the re-emerging, we also then found something emerging. For the first time here, another coffee leaf spotter called Paramyrothecium roridum, which was found in more than one location in the field. That's what these pictures are here, and particularly seems higher incidence and severity in the greenhouse. And these are what the pictures that you're looking at here. And so this was one of a first report for Hawaii. This is what I'm talking about. This is the field. This is the recreating of it artificially, completing pathogenicity Koch's postulates, what the fungus looks like, both growing on the plate and under the microscope. And this does exist around the world. It is known more for being causing more problems in a greenhouse. And so we are getting a better handle on this pathogen and doing some fungicide screening for management. Okay, I talked about coffee leaf rust. One of the major areas is variety screening, and the team developed an optimized leaf disc assay so we could do screening fast with everything we could get our hands on, all the material existing in greenhouses, even in the field, collecting leaves, making leaf discs, doing testing. We started small, we've ramped up. The team's amazing how much it has gone through. Probably with HARC accessions and the university, we're talking close to maybe 1,000 over the last couple years. This is what we're looking at and for. So this is a very susceptible reaction on these leaf discs, and this is a close-up picture of all these coffee leaf rust spores. And it's a high concentration that we're putting on the leaf discs. And what happens sometimes is you will see a chlorotic spots, but spores don't, they're not produced. So the pathogen is not completing its life cycle, which is great. And so that's it. It gets in, it causes some localized damage, but it dies. And then there's many examples actually that we have now that we don't even see spots. So when we looked at, like I say, close to 1,000, 76% is showing great resistance or tolerance. So a lot of good material prioritized to do potted plant and to help work collectively to get this material to growers. Okay, another aspect is biological control. So we noticed right away working with coffee leaf rust that there were fungi attacking the fungus. And we did a huge survey, got, oh, this is what you're seeing, all the diversity planted, doing testing to see how maybe some of these natural things occurring can actually halt disease. Okay, whew, sorry, I know that's like, whew. I'm going to touch upon soursop because this is, in a way, the latest major problem. It seems throughout the state. It's very, very bad on Hawaii Island. Some of these symptomology, and it's not just soursop, it's Annona species, Rollinia, sour sugar apple, Annonas. And you see some of this discoloration. You see a lot of curling, twisting, small leaves. And the one thing that we've noticed, and we have sampled all over, and have also gotten samples from Maui, same kind of symptomology, and this little pest right here. These leaf hoppers, and just to show you, these are probably like, I don't know, 25 leaf hoppers in a tube. These are my fingers. They are very, very tiny, but they seem to be wreaking havoc on our crop here. And what was very interesting is we had started just to do some pathogenicity testing if and when we found fungal or bacterial potential pathogens. We had some seedlings growing and we put, so this is outside our shade house, a sample subsample of them actually in just a very simple to prevent insects and not. So right next to each other on the bench. And what we started to observe over time under the shade cloth, insect netting, they're growing quite nicely out in the open with potential bugs, really other things. You could see what's happening. We actually just started to see the symptoms develop that we were seeing in all of these fields. Okay. We've done extensive isolations and testing, and this Annona mystery continues, but we're thinking we have a much better handle of it now because we can say a whole lot of what it is not. It is not phytoplasma. It is not Xylella. We do not suspect any major new fungal or bacterial pathogens. We did find something interesting in the soil at one location, but not all over. We'll be looking at that separately. It is possible that it is an abiotic thing with the temperatures. I mean, it seems like a lot of rain, but maybe the water is not really getting to the roots, but we find leaf hoppers everywhere. And we've also tested the leaf hoppers to see if they as potentially are vectoring something, which they do not appear to be. A couple things. Florida is suffering from this same issue that this is a picture from a study there. Exactly the same kind of symptomology, discoloration in the leaves, the curling, the small, lots of leaf hoppers. It then doesn't take a lot to actually start causing a problem and almost an irreversible problem with too much feeding. I will say that thinking, well, could it be of the pathogen? We knew we started clean when we moved the diseased looking seedlings inside a protected greenhouse. The seedlings actually then grew out of the symptomology. The new leaves started looking good. So we really do suspect that this is the culprit. And from there, summary, plant diseases are difficult to control. there is no silver bullet. Everything is about integrated pest management and hopefully reducing disease to an acceptable level. Here are some of the things you can do. And then this is the type of work that my team does and will continue doing to try to get as many tools for the growers to help combat these problems. And with that, I will say thank you very much. I appreciate your time. If you have any questions whatsoever, please reach out. Here's my email, and I'll try to answer any questions now. Thank you very much.